to make a start, even if I'm talking to myself. You're not talking to yourself, otherwise we'll never get home. So we're going to talk about necrotizing enterocolitis. So I have done a talk similar to this before, but I've incorporated quite a lot of new stuff into this talk. So Gerard, believe me, I haven't done this talk for you before, but you have heard a neck talk from me. It's not this one.
Not anyway.
Okay, so those of you who read journals might know that there's been an absolute explosion in evidence about NEC, which is just as well, because really for many, many years we were completely stumped as to what causes this awful condition in babies. Have any of you trainees seen babies with NEC? Can you stick your hands up if you have? Have you never seen? You've never seen? Okay. Can you briefly tell us what happened, if you can remember?
So the baby that sticks in my head the most was an X25 weeker, I think, who got to about 32 weeks.
Yeah.
And he just suddenly became very, very unwell. His belly was quite distended, discoloured. He wasn't perfusing very well. Got intubated, had surgery, and then died post-operatively.
Yeah.
He was quite poorly.
Yeah. I mean, that really is the parents' worst nightmare, isn't it?
Yeah.
You know, the baby that seems to be doing well, and then several weeks down the line gets really, really sick, and despite all our efforts, succumbs. And you know, this is the tragedy of necrotizing enterocolitis. It's a shocking condition, and one which has eluded really prevention. So this is what I'm going to talk about. So this is an example of a belly of a little baby with necrotizing enterocolitis. So these are babies that, sort of babies that are usually very prem. They're on our unit, and then, you know, a few weeks down the line, will develop a distended abdomen, which can be discoloured, can be red and shiny, tense and tender. There may be bilious aspirates, there may be blood in the stools, and the baby gets very acidotic and shut down and really unwell. The treatment for the condition is medical in the first instance, so it's supportive management, draining the nasogastric aspirates, nil by mouth, and antibiotics, and then surgery for if they perforate. And the results from surgery are that probably somewhere like half of them will die despite surgery. And some of those who don't reach surgery will die as well, because they just get so overwhelmingly unwell that you can't really move them to a surgical centre. So this is another baby with a distended abdomen, as you can see. And if you saw a baby like that, you would seriously wonder about perforation there because of the amount of distension and discoloration of the baby's abdomen. Now, can we just turn the lights down a little bit, please, because I don't know whether you can see that otherwise. So, can you see this abdominal X-ray, and can you point out to me what is abnormal on it? So this is what you're looking for, really, if you do an abdominal X-ray in a baby with suspected NEC. So can you see, who would like to comment on this? So what you can see is this sort of bubbly pattern in the gut wall, which indicates gas. Okay, so this is gas that's produced by gas-producing organisms. It's actually hydrogen gas. And you see this, this is one of the characteristic radiological features, and this is what we were actually looking for with necrotizing enterocolitis. And it's a relatively late sign. So an awful lot has gone on before you get to this stage that leads up to a NEC. Now this is some histology. So the first one, anybody like to hazard a guess as to what that shows? Which bit of the intestine is that, do you think?
Small
intestine. So you've got villi, you've got crypt. This is a normal small intestine, okay? So you've got a single layer of cells here with lacteals on a connective tissue base. So what's happening here? This is necrotizing enterocolitis. What has happened here?
Well, you've lost your individual villi. It looks like they're broken down.
Absolutely. That's exactly what happens. You lose the integrity of the villi, and yes, you're right, it's all broken down, it's all disrupted, there's hemorrhagic necrosis and inflammation. Sometimes you can even see bacterial cells in there, and this is an even worse case. So as you can see, what happens here is the epithelial layers just literally fall apart, and this leads to the intestine perforating and the abdomen distending, and they get very difficult to ventilate. It's a very painful condition, and yes. So what's happening that makes the gut get to this stage? So NEC's been known about for many, many years, and it's not just humans that get it, but other animals. So it's recognised by vets, and it's known in experimental animals as well. Originally, it was thought that there were 3 mechanisms leading to it. So intestinal ischemia, followed by colonisation by pathogenic bacteria, and usually associated with not human milk feeding, but formula feeding. That was the original concept of it. There are some interesting facts about NEC. It's never seen in stillborn infants. It's very rare in babies who have never been fed, although it can occur in babies who've never been fed. And it's predominantly those very small babies who get NEC. So it's the baby, just as you described, the typical preterm baby, a few weeks down the line, suddenly hits it out of the blue. Nowadays, the most accepted sort of precursors are extreme prematurity, gastrointestinal feeding, formula milk. We've seen this ourselves many times, where babies are given mother's milk or donor milk. You get to 30 weeks gestation, mum's milk dries up, right, we give formula feed, next day or 2, NEC, and it's absolutely devastating. Now this is a graph showing the distribution of the gestations of the babies who get NEC. So if we just ignore the SIPs for now, which is a different condition, spontaneous intestinal proliferation, ignore that bit. But if you look at this, you see these are the NEC babies. You can see that it's primarily this group of babies.
This is gestation at birth rather
This
than
is
the gestation.
This is gestation at birth. So, and you can see for surgical NEC, you know, there's quite a lot of babies. It's about nearly 50% of these babies requiring surgery. And as they get sort of more towards term, fewer babies actually require surgery, fewer proportions of babies. But it's rare for a 23-weeker to get NEC on the first couple of days. They normally get it several weeks down the line, as we've said, whereas the term babies may get NEC on day 1 or day 2. And if you look at the corrected gestational age at NEC onset, you get this thing called the SART-12 chart, and you can see that it's around 31 or 32 weeks that it happens most commonly. And this is why we've had on our unit, in our guidelines, that we will really try very, very hard only to give mother's milk or donor milk up until 34 weeks. 34 weeks. Okay, and if we do that, we hope that we will get our babies through. There's, you know, one or two we might miss, but on the whole, since we've been doing that, touch wood, we believe we have seen fewer cases of NEC, so that may have done some good. When trying to work out what causes NEC and how we can prevent it, people have thought, well, actually, maybe it's not all one condition. The commonest scenarios, as we've described, the very preterm baby getting to 30, 31 weeks, but there are other scenarios as well that people talk about. There's the thing called transfusion-associated NEC, which is where a baby develops NEC within 48 hours of a blood transfusion. This may not exist as an entity, but people wonder about it. Personally, I don't think it exists. However, lots of people have written papers about it, and I think
Lots of people still withhold fees around.
yes, they do. Then there's a thing called, rather funnily, moo-neck, which is probably associated with cow's milk protein allergy. So this tends to be more the term babies or late preterm babies who are given formula feed and seem to get a bit of a bloody stool and feeding intolerance plus an eosinophilia, and we tend to cure those by putting them on a cow's milk free formula. Then there's a thing, NEC plus known bacterial pathogen. When I was a registrar, we had an outbreak on our unit of NEC. You know, there were lots of babies going off with NEC, and it seemed to be associated with C. diff infection. Hopefully we wouldn't see that. I suspect our infection control practices weren't that brilliant on that unit at that time. Enough said about that. NEC plus known viral pathogens has also been identified, and then you have NEC in your HIE group of babies. There's cold stress NEC, where you get NEC in babies who failed weaning from an incubator because they've been cold within the last 72 hours. NEC in gastroschisis babies, NEC plus a known genetic variant, I'll say more about that in a minute, NEC plus a congenital abnormality such as congenital heart disease, and NEC plus a commercial product. And we've identified on our unit a couple of things. So Carabel and Gaviscon, which are known to be associated with NEC, hence we avoid them in our preterm babies. So all of these things may be slightly different conditions, resulting in the final end common pathway. So, as we've said, it seems that NEC is associated probably in most babies with an immature immune system. The 23-weeker who gets NEC around 30 weeks. It seems to be something to do with aberrant colonisation of the GI tract. Certainly it's got something to do with mucosal inflammation. There may be predisposing factors such as genetic factors, inflammation, which can be pre or postnatal, lack of antenatal steroids predisposes, increased antibiotic use, formula feeding is a biggie, but underlying it all, it's that integrity of the intestinal mucosal barrier which is broken. And there's a whole pathogenesis associated with this, which involves the immune system, both the innate and the adaptive system, which are associated with this. So if we look at why preterm infants may be particularly predisposed to NEC, what have we got? So here's our intestinal epithelial layer. We have on the surface of it, we have cilia. We also have a layer of mucus and a layer of proteins, okay? Now, in the preterm baby, these things are very immature, and so whereas you should have a bit of a no man's land between the surface of the epithelial cells and the the bacteria in the gut. In the preterm baby, this is much closer together. Also in the preterm baby, the tight junctions between the cells are not well formed, so these can be more permeable. In the stomach, instead of the stomach being very acid, that isn't the case, so therefore bacteria that are ingested aren't killed as much as they are in the term baby or the child or adult. The colonisation of the gut in the preterm baby is different from in the term baby. So that may well be having something to do with it. And we also have immature mucosal immunity. So what is thought to happen is that for some reason bacteria can penetrate between the cells, set up an inflammation. This stands for platelet activating factor, which then leads to an uncontrolled inflammation and leads to apoptosis of the cells and breakdown of the mucosa. So that's some of the reasons why the preterm mucosa might be more susceptible. Here's another slide, and what this shows is things called toll-like receptors, which are on these cell surfaces. Toll-like receptor 4, TLR4, is particularly important in the pathogenesis of NEC, and people have shown this. TLR4 binds with lipopolysaccharide in gram-negative bacteria, so things like E. coli, and that sets off a huge inflammatory cascade. These things here are sort of holding back that inflammatory cascade, and these can be reduced in preterm babies as well. So the inflammatory cascade, TLR4 activation, leads to increased apoptosis of the cells, decreased restitution. So the epithelial cells, they're formed in the crypts, where there are the Paneth cells and the goblet cells. They're formed in the crypts and they move up the villi, and then eventually they die and they fall off. But in NEC, this process is very slow and the cells don't proliferate. There may also be decreased Paneth cells and goblet cells in the preterm baby, which again predisposes. So there are all these complicated factors which kind of line up, a bit like the holes in the Swiss cheese, which predispose the preterm baby to NEC. Now, there are interesting animal models of NEC. People have done experiments on, for example, rodents such as mice. And if you give a pregnant mouse lipopolysaccharide, which can be either intravenously or into the amniotic cavity, the baby mice that are then subsequently born are inflamed. So they have increased inflammatory markers, IL-6, 1-beta and 17. If you keep those baby mice with the mother mouse and they're fed her milk, they don't get NEC. But if you remove them and give them a formula feed, they get NEC, and that's a good animal model. They can also make these mice with absent TLR4 receptors. So if you do the experiment again in the mice with the absent TLR4 receptors, they don't get NEC, which kind of proves that TLR4 receptors are very important. And there are various other experiments that they've done as well, showing how different components of the immune system are very important in the etiology. They think that this is all applicable to humans as well. In humans, they've looked at various genetics in infants who have NEC and have shown certain genetic polymorphisms which predispose to NEC. There's a thing called SIGA gene. I've forgotten what it stands for, but essentially it's one of these things that holds back that inflammation cascade involving the TLR4 receptors and lipopolysaccharide. And there are other things which also predispose to NEC and things which can be protected as well. So there's individual variation in susceptibility. And there are some unfortunate families where you know, several babies in the same family will get NEC, which is very unfortunate. Just a little bit about the gastrointestinal motility. In preterm babies, this is immature. One of the things about gastrointestinal motility is that the microbiota, the microbiomes living and residing in the gastrointestinal tract produce a number of metabolites, and these include 5-HT, which is important in gastrointestinal motility, surprisingly, and also important in the maturation of the enteric nervous system to do with peristalsis. So it may be that deranged microbiota may adversely affect gastrointestinal motility, and then that may lead to a bacterial overgrowth of the unfriendly or hostile pathogenic bacteria. Poor peristalsis will also increase the time that antigens are able to interact with the enterocyte. And luminal digestion is really the first line defense against this. So it could be that a combination of poor motility and immature intestinal digestion again predisposes the infant to colonization by these abnormal pathogens. So just to recap then, prematurity, formula feeding, the use of antibiotics by causing a deranged microbiota, these all predispose. We've talked about barrier dysfunction and a number of immune factors, toll-like receptors 4, and so on. Altered growth, again, and there are various cytokines which are involved, and I've listed them here. So it's very, very complicated. The whole immunity is extremely complicated and still being elicited and worked out, with the hope that ultimately maybe that some of these low factors may be able to be replaced and maybe prevent or treat NEC. So other things then that may predispose the preterm baby, we've talked about gastric acid, that is, well, the gastric juices are not so acid in the preterm baby as in the term baby. That may be one of the things that predisposes the baby. The mucus layer in the preterm baby is thin and not very effective. The epithelial layer, the tight junctions are not well formed. They mature at term. And also there are various antimicrobial peptides which are produced by the term gut, which are not very well produced in the preterm baby. What else? Well, we know that in intrauterine life, the mother who has a lot of IgG on board, this will pass through the placenta into the baby, and the nearer term the baby in utero approaches, the higher the IgG level. So babies who are born at 23 weeks, 26 weeks or whatever won't get that dose of maternal IgG, which helps to protect them from infection. Against that, breast milk transfer of IgA. IgA is an important factor which helps to protect babies. And we know that it's a good thing to breastfeed babies, but fortunately in preterm breast milk IgA is quite well produced, so that is one thing that sort of helps us. Why is human milk so good for babies? Well, one of the things is that it contains a whole load of protective factors, including oligosaccharides, caseins, triglycerides, lactoferrin, and lysosome. I'll talk a bit about these in a minute. And also it's got a number of immunoregulatory cytokines, growth factors, etc. So let's just go on quickly. So this is just to recap on the way that human milk protects. It's an easily digested protein. It decreases intestinal permeability. It's anti-inflammatory. It promotes a beneficial intestinal flora. It contains oligosaccharides, which are food for the friendly bacteria. So that's the bifidobacteria in particular. And it reduces inflammation. Acetyl hydrolase is one of the things that sets off inflammation, so it reduces that and it prevents bacterial invasion. It also matures various components of the immune system. So very, very highly complex. In fact, it's amazing. The more you look into it, the more absolutely incredible it all is, really. I won't go into that. So just to say about a number of studies that have been done, these are quite old now, comparing preterm babies fed formula to breast milk, and a much higher incidence of NEC in those who were fed formula. And the benefits of human milk are dose related, so for every 10% increase in milk, there's a reduction in NEC by 0.83. So it's undoubtedly one of the key things. Looking at feeding advancement, and we had the SIFT trial, which we did on our unit, where we compared a rate of increase in feeds of 18 mils per kilo per day with 30 mils per kilo per day. This was because people thought that increasing feeds faster may predispose to NEC. It showed that there was absolutely no difference. And in fact, if you increase slowly, then all that happens is that babies don't gain weight so well and may get more infections because of long lines. So it's a good thing to get the babies feeding 30 mils per kilo per day, starting with breast milk as soon as possible after birth. Continuous versus bolus feeding, there's no difference really. Okay, so I'm just going to talk a little bit about bacterial colonization now. So it used to be thought that in utero the fetal gut was sterile. Nowadays people think that that may not be quite right, and in fact the intrauterine environment, even in healthy pregnancies, may not be absolutely sterile. Now, where do the germs come from? Well, there are again a number of theories. One of them is that it comes from the mother's oral mucosa, which can result in a low-grade bacteremia, and these can then seed into the baby. Other theories are like the pathogens or the microbiota comes from the vagina. And other theories are that it involves the dendritic cells, which are components of the innate immune system, which can, these are mobile cells, so they can sample the microbiota in the mother's intestine. It's a bit like sort of sticking a finger up in the wind, it's finding out what's there, and then travel to the baby in utero and either prime that baby's immune system or seed that baby with those particular organisms. I don't think they quite worked out exactly what's happening. So this is a little bit about the microbiota composition. So these are term babies compared with preterm babies at 2 days, 10 days, 30 days and 90 days. So these are the term babies, these are breastfed term babies. You can see that the microbiota does change over the first 3 months, but on the whole is pretty static, and the predominant things, the predominant organisms here are the bacterioid ACI and the Enterobacteriaceae. And in the preterm babies, they have a slightly different spread of organisms. So these are quite different, especially in the early in the early days. But later on, the microbiome more closely approximates those of the term babies. And this is important because the microbiota is the thing that primes and develops the immune system of the baby. Perhaps I won't go into that now. It's a bit complicated, but essentially term breastfed babies have, within a few days of life, developed a normal gut flora, bifidobacterium and enterobacteriaceae. Bottle-fed babies of a term are colonised with enterococci and bacteroides, and preterm breastfed babies are colonised by a narrow range of bacteria with lactobacilli and bifidobacterium, which are the friendly bacteria, accounting for less than 5%. So preterm babies do not have what we would call a healthy microbiota. In babies who have NEC subsequently, people have studied what happens to the microbiome, and what they found is that the microbiome is quite unstable and changes from day to day. Some people have said that certain groups of bacteria are very common in babies who get NEC, but when you look at the studies, they all seem to differ in respect to what particular bacteria there are. So I think that the take-home message that I get is that the microbiota of the preterm baby who gets NEC is very, very unstable and rapidly changing, but I don't know that any particular bacteria predominate. So this brings me on to prebiotics, probiotics and postbiotics. Okay, so prebiotics. These are mainly the oligosaccharides, which are components in human breast milk. These are complex sugars ranging from 2 to 10 sugars in a chain, and they can be glucose, galactose, glucosamine, fucose, or yes, that's right, or fucose. And they can have side chains on them. So any mother's milk will have 100 or more of these different oligosaccharides, which are quite specific to her. And as we've said, that these are specific foods for friendly bacteria. So these are the bifidobacteria and the bacteroids. Now, formula milk companies claim to put oligosaccharides in their formula, but they only put 2 oligosaccharides, neither of which has any recognisable function in humans. So it's a big gimmick and we shouldn't believe it. The only way that we can give these oligosaccharides to our babies is to give it in mother's milk. Even the donor milk that we use will have oligosaccharides in it because these are not destroyed by pasteurisation or freezing. So it may be that the benefits of donor milk come from that. Now probiotics, on the other hand, these are live organisms which, when administered to the baby, confer a health benefit. These are live organisms, and ideally they will have gone a controlled evaluation, and it's a taxonomically defined microbe or combination of microbes safe for intended use. So there are a number of probiotic bacteria which include lactobacillus, bifidobacterium, bacillus, saccharomyces, Escherichia and Enterococcus. And I'm just going to talk about 2 of these. Within the individual families, there are a whole sort of host of subspecies. So starting off with the bifidobacteria, these colonise the intestine in term infants quite commonly, and they can consume the human milk oligosaccharides. The sort of strains of this, there's one called Bifidobacteria infantis, and this can consume all the types of human milk oligosaccharides. They can also produce a single bactericidin, which will have activity against staph, strep, salmonella and E. coli. And in rat models, this particular probiotic decreases NEC and decreases the expression of various inflammatory markers. Another substrain of bifidobacteria bifidum, this can produce 2 known bactericidins and also produces more short-chain fatty acids than other strains. So what you can see from this is that these different substrains work in different ways. Not all probiotics work in the same way. So this idea that they're friendly bacteria, they just fight off the bad bacteria, and it's a question about numbering them, is very much oversimplistic. These are very complicated things, and it's really, again, quite amazing how they work. So moving on now to the lactobacilli, these are not typical colonizers of the newborn GI tract. They can't consume the human milk oligosaccharides. Looking at a subspecies, Acidophilus, these can produce at least 27 different bactericidins with a wide range of activities. They also secrete anti-inflammatory factors inhibiting the induction of certain inflammatory mediators, and they also produce short-chain fatty acids and factors that facilitate the uptake of butyrate by host colonocytes. So this is important for the health of the enterocytes in the colon. And there are lots of other different strains which act in different ways. So one of the problems with the probiotic trials that have been done is that they've all used different probiotics, and because they work in different ways, they may not actually be comparable. So there have been many different trials of probiotics in preterm babies looking to see if they can reduce NEC infections and death. One of the earliest meta-analysis that was done was one by Deshpande et al., and that was really calling on neonatologists to use probiotics in preterm babies. There have been lots of more recent meta-analyses involving more recent studies, and again you can see that the results are pretty consistent in that they reduce the NEC rates by statistically significant reductions, approximately a half the rate of NEC. The rates for death, again, most of these meta-analyses show statistically significant reductions, with just one where it crosses 1. So really, a very large body of evidence stating that if we give the babies probiotics, that this will reduce NEC. There is also some of these meta-analyses have looked at the reduction of late onset sepsis. The reductions aren't so great. So it's really in the reduction of NEC, and a wide range of different probiotics have been used in these.
Can I ask, is the Elfin trial a probiotic?
No, that's lactoferrin. I'm coming on to lactoferrin in a minute. So why aren't we using probiotics? Well, people have been quite reluctant because of concerns they've had. So there have been very, very occasional, probably about 3 or 4 cases of sepsis with a probiotic organism in preterm babies, which has put the Kybers up everybody, not surprisingly. There's also been an incident where one of the products, Acidophilus, which is a probiotic, a manufactured probiotic, was contaminated with a fungus, and this affected some babies who developed very nasty infections, and I think one of them died. Because probiotics are classified as foods and not drugs, it means that there is not a quality assurance about their manufacture, so that different products may vary in the viability of the organism and even the type of organism in the product. So it may be that you're giving something that you don't really know what's in there. You just hope for the best. And there's also evidence from trials that these probiotics, if you give it to, if you do a placebo-controlled trial, it's not just the cases who get the probiotic, but somehow the controls seem to get it as well. So there's cross-spread against the urine. How that happens is anybody's guess. Whether we should be concerned about that, I don't know. So,
Jean, is there, I've tried to look for this before, but is there much evidence that if you supplement mothers with probiotics, that has any effect on NEC rates?
there isn't, I've not seen any trials on that, but I've seen it as a suggestion.
Yeah.
And that may be a way forward.
Because that would be something really easy to do,
Absolutely,
wouldn't it?
absolutely. But I think even
so a mother's, just before they deliver and in the weeks to months after while they're expressing.
I think there has been a study, but not looking at NEC rates. It wasn't designed to look at NEC rates. But what it did look at was, and then supplementing the babies as well. These were term babies, looking at things like allergy later on. So that's been done, but not specifically designed for NEC. It would be easy to do. I have to say one thing as well, that in Canada now, they have a much more stringent manufacturing process for their probiotics, and there's one called Flora Baby, where there are 5 probiotics, and it's subjected to the same sort of manufacturing standards as if it is a drug. So there's much more quality assurance about what they're giving, and I think if we were considering giving probiotics, that's the one I would suggest we give. I'm not entirely convinced we're doing the right thing by not giving them. I understand the concerns. We'll see. Lactoferrin then, so this is in answer to your question. This is the trial that we did, called the ELFIN trial. So lactoferrin, this is a major whey protein in maternal milk. It acts in a number of different ways to protect the baby from infection and, we hope, NEC. It's a naturally occurring substance. It has a role in intestinal growth and maturity after birth. It works with lysozyme to kill enteropathic bacteria. It's anti-inflammatory. It matures the T and B cells. It binds endotoxin, and it kills bacteria, fungi and viruses and parasites, and it inhibits biofilm formation. So these are all things that make us think it should be jolly useful in preventing NEC in babies, which is why we've just done the placebo-controlled trial, which has just finished. And it'll be very interesting to see what that shows. But meta-analyses that have been done on the use of lactoferrin have shown very encouraging results. So it may be that will be something in the future for us as well. So just a little thing about volatile organic acids. The microbiome in the gut is a metabolically active organ. So all these different bacteriological species that exist in our guts are performing vital biochemical functions. Thousands of them. I mean, the genetic material in the microbiome by far outnumbers our genetic material. And from producing things like neuromediators, short-chain fatty acids which keep colon cells healthy, dealing with toxins, the list of what they do goes on and on and on. Now it's very interesting that we can measure in stool things called volatile organic acids, which must be substances which are produced by the microbiome. We can measure them by some sort of device called an electronic nose, so it can pick these things out. And I imagine that, animals like dogs would be very good at picking these things up. However, there we go. What they have found is that when you look at very low birth weight babies and you serially measure volatile organic acid compounds over the days, the babies babies who went on to develop NEC had a different group of volatile organic compounds than those who didn't, and these were detectable a few days before they actually came down with NEC, implying something about the microbiome of the baby. Something was changing. These compounds of the babies who got NEC were also different from the volatile organic compounds of those babies who got sepsis. Those who got late onset sepsis could also be discriminated from controls without sepsis at 3 days, 2 days and 1 day prior to the onset of sepsis. You just wonder what on earth is going on here. But is this a tool that we could be using in order to detect what babies are going to get NEC and what babies are going to get late onset sepsis, which would then allow us to intervene? Well, that would be absolutely fascinating and fantastic if we could do that. But the numbers of these trials are small, and I think more information is required. A little bit about NEC at Singleton. This is quite an old slide now. I looked at the incidence of NEC over a 5-year period. It was 5.5% of our very low birth weight babies, compared with the Vaughan average of 7%, so we were a little bit less than the Vaughan average. And of the 22 babies, we had more than half transferred to surgical centres, 10 had surgery, 5 died. I don't know if I dare say this, but I think over the last 2 years, since we've been reducing antibiotic usage and really stepping up the breastfeeding, I think we may have seen 1 case of NEC, which was a milder case which got better quickly. But this could be a blip, it could, because it is a condition which does this, so we could just be in a good phase, it could be coincidental, it's a bit too early to say. Let's hope though that it is a trend. So our statistics are pretty similar to the rest of the Vaughan network back in 2005-2009. That's just a summary slide about what predisposes to NEC. I went to a conference on NEC about a couple of weeks ago, and there was a lot of talk about immunology. It was really very detailed, but it kind of just gives you the flavour of what's going on. I can't say that I understood absolutely everything that was being talked about, but there are some hopes of new treatments. IL-22 recombinant as a treatment for babies with existing NEC, that is being put to the FDA in America for approval as we speak. So that may be approved it works in experimental animals. They're looking at probiotics targeted in nanoparticles, which would mean that the probiotics might get to the gut better than they do now, where at the moment probiotics have to ride the gauntlet of the gastric acid in the gut and get to where they need to be, and then set at home, as it were. So it may be that having them targeted in nanoparticles may help. They're also looking at tissue engineered gut, using stem cells, growing gut from a baby's stem cells, which they can use in short gut syndrome. And they're also looking at setting up a biorepository, a bit like what was envisaged for the baby biome study. So with babies collecting urine, stool, breast milk, cord blood, sticking it all in a biobank, and then sort of seeing who gets NEC, who doesn't get NEC, and then seeing what's going on, what's the difference in the immune factors, what's the difference in the microbiota, what can we learn from that. So that is happening very much in America. It's in individual centres, but they are all collaborating, so maybe something good will come out of that. So I think the key messages are that it's a devastating illness. We, until recently, haven't really had much of a clue about what's going on to cause it. People are now beginning to get some of the pieces of the jigsaw. But the main messages are that human milk is really the key feeding strategy to prevent it, and that we also need to reduce our antibiotic use, both for the mothers and the babies, probably, and that will help as well. So that's it. That's me done. Great. Thank you for your attention.
Thank you. Can I have one question?
Yes.
It's just because I didn't go to the lecture, I was taught there's quite a big deal with regards to risk factor for NEC. Is that the sort of reduced gut flow, endosmotic flow and things, is that still a really big factor? Or is that less so now that more of this is coming out?
People have looked at that factor, and they've looked at differences in feeding. It does, how you feed them doesn't make any difference, so you don't have to give them slower feeds.
That's what we do in Carlo times. If they're at a low risk, we really start with slow feeds.
No, you don't have to. There's no evidence that it makes it worse.
Okay.
I wonder whether it's worth looking back at our NEC babies and seeing whether these mothers had inflammation beforehand. You know, just like the mouse model I mentioned, where giving the mouse, pregnant mice, the lipopolysaccharide, which then induced the inflammation in the babies, which then kind of made them more prone to getting NEC. I wondered if that's something similar is going on.
The only thing you're going to get with septic models is the only people you get pick up with inflammation, isn't it?
Yeah, septic or maybe some
autoimmune.
Yeah, I don't know. Our numbers are too small really to make much headway with that. But there we are.
That was excellent.
Yeah, thank you very much.
Excellent.
I wish I understood immunity, but
Yeah.
thank you.
Thanks, Jules.
Sorry. Steph, never mind, I forgive you. Thank you all anyway
Thank you.
for your attention.
Yeah, every now and again it's nice to a real sciencey one, isn't it? Every now and again.
Was that a real science?
Yeah, oh yeah. I
Well-
this and that, and trottle this, toll this and that, and
Was that being
flashbacks and
taped?
vegetables. I don't think.